Morning. So yesterday we studied about the different basic components of cell, and the basic components of cell include plasma membrane, nucleus, and the cytoplasm. In that we studied about the plasma membrane. We already studied about the plasma membrane. Next we have to study about nucleus in detail. We know nucleus forms the control center, main control center of a cell. Nucleus is the main control center of the cell. It controls all the activities of the cell and also helps to carry the genetic information from parents to offspring. So we will study about the nucleus and cytoplasm and the cell organelles which are present in the cytoplasm in detail. So today we will study the topic nucleus. Nucleus is the main control center of a cell. When we consider nucleus, nucleus is the largest organelle and it forms the main control center of the cell. Nucleus is actually a dense and spherical organelle which is present in the cell. So now first we see the structure of a nucleus. When we see the structure of a nucleus, it has a outer double layered membrane. The outside, it has an outer double layer, two layers are there. Double layered membrane is there, and double layered membrane is called as nuclear membrane or nuclear envelope. So the outer double layered membrane of a nucleus is called as nuclear membrane or the nuclear envelope. And these nuclear membrane has got so many pores in it. Pores are there. What may be the function of these pores which are present in the nuclear membrane? These pores actually helps in the transport of materials in and out of the nucleus. It helps to transport materials from inside the nucleus to outside the nucleus and also to carry certain materials towards inside the nucleus. So the nuclear pore which are present in the nuclear membrane helps in the transport of material in and out of the nucleus. So that is the second part. Coming to the third part, that is nucleolus. Nucleolus is a round, spherical, dense region which is present inside the nucleus. And this nucleolus has certain functions like helps in the synthesis or the production of ribosomes that you will be studying about that detail in the coming sessions. So nucleolus is a round, spherical, dense region which is present inside the nucleus. And within the nucleus, we can see a jelly-like fluid like cytoplasm. Inside the nuclear membrane, we can see a jelly-like fluid called as nucleoplasm. Nucleoplasm is the jelly-like materials which is present inside the nucleus. And in the nucleoplasm, there are tiny, small, long, thread-like materials called as chromatin materials are present. These chromatin materials are the main genetic material which helps to carry or helps in the transmission of genetic characters from parents to offspring. So this is about the basic structure of a nucleus. Structure of a nucleus include, we know nucleus is actually a dense spherical organelle. It has several parts. Outer double layered membrane called as nuclear membrane. Then the nuclear membrane has got nuclear pores in it, and the nucleolus is the dense round region which is present in the nucleus, and jelly-like fluid which is present inside the nucleus is called as nucleoplasm. And inside the nucleoplasm, the chromatin materials are floating. Chromatin materials are the genetic materials which helps in the transmission of characters from parents to offspring. Now we study about this genetic material in detail. So now we will study about the genetic material in detail. We studied genetic material which is present inside the nucleus, that is called as chromatin material. So what do you mean by chromatin material? Actually, this chromatin materials are thread-like materials which carry genetic information. Chromatin materials in the diagram we saw, these chromatin materials are thread-like materials which helps to carry genetic information. And during the time of cell division, or when the cell is about to divide to form two cells, we studied in the cell theory Virchow proposed that all cells arise from the pre-existing cell. So when a cell is about to divide, that time what happens? These chromatin materials, these long thread-like chromatin materials will condense to form a solid structure that is called as chromosome. These chromatin materials condense to form the chromosomes. And chromosome contain the genetic information. When the cell divides, this chromosome contains the genetic information that will be passed to two newly formed cells. And now we see the constitution of chromosomes. Chromosomes are actually made up of DNA and protein. It is important, the constituents of chromosome, it is DNA and protein. Chromosome is made up of DNA and protein. What is DNA? DNA is actually a biomolecule, which is expansion of DNA is deoxyribonucleic acid. It's a biomolecule. It's very important biomolecule which contains nitrogen, phosphate and sugar. All we will be studying about DNA in detail in higher grades. So chromosome consists of DNA and protein. So these are the two basic constituents of chromosomes. And when we consider DNA, it has got functional segments, and the functional segments of DNA is called as gene. Gene forms the functional segment of DNA. These are all important questions for 1 mark. Chromosome constitute DNA, it's actually made up of DNA and protein. Expansion of DNA is important, that is deoxyribonucleic acid. DNA is actually a nucleic acid. So expansion is deoxyribonucleic acid. And gene forms the functional segment of DNA. DNA has got thousands of genes present in it, and each gene code for the character, particular character like color of hair, color of eyes is actually coded by a gene. It's actually a segment of DNA. So this is about the genetic material which is present inside the nucleus. Now we will study the functions of nucleus. Nucleus, we know nucleus actually forms the main control center of the cell, and it carries the genetic information which has to be transferred or transmitted to the offsprings, from parents to offsprings. Now we see the two main functions of nucleus. One is nucleus plays a central role in cellular reproduction. What do you mean by cellular reproduction? Cellular reproduction means obviously the reproduction of cell. Reproduction of a cell means single cell will divide to form two new cells. So the nucleus controls the cellular reproduction, the cell division, the time of cell division, everything will be controlled by the nucleus. So the nucleus plays a central role in cellular reproduction. Second function is, along with the environment, nucleus will determine the way the cell develops and what form it exhibits at maturity by directing the chemical activities of the cell. So by directing the chemical activities of the cell, it controls, by controlling the various chemical activities that is taking place inside the cell, the nucleus can determine, along with the environment. Environment also plays an important role in determining what form a cell should occur when it matures. So along with the environment, the nucleus will determine the way the cell develops, how the cell should develop, and what form it should exhibit. The cell, once a cell is formed, it will have to get matured and it have to attain its particular function. So that will be controlled by the nucleus of the cell. So along with the environment, the nucleus will determine the way the cell develops and what form it exhibits at maturity by controlling the chemical activities of the cell. It helps to control the chemical activities of the cell, thereby determine the form of the cell, and the function, what function it has to do, will be determined by the nucleus. So these are the main functions of nucleus. One is, it helps in cellular reproduction, that is, once a single cell divides to form two new cells. And second one, it determines, along with the environment, it determines what form the cell should occur and what should be what way it should develop during the time of after cellular reproduction. The cell has to acquire the form and function. So that will be controlled by the nucleus by controlling the chemical activities of the cell. Now we will see the types of cell based on the type of nucleus. How define the nucleus? Based on that, there are two types of cells, and they are actually prokaryotic cell and eukaryotic cell. From the term prokaryote, pro means primitive, karyota means nucleus in Latin. So pro, primitive, karyota, nucleus. So prokaryote means with a primitive nucleus, and eukaryote means eu means here true, and eukaryote means with a true nucleus. So from the term itself we can get the definition of these two types of cells, prokaryote and eukaryote. Prokaryote means a cell without a well-defined nucleus, with a primitive nucleus, there the nucleus is not well-defined. And eukaryote means with a well-defined nucleus. So we will study about this prokaryotic cell and eukaryotic cell and the examples of prokaryotic cell and eukaryotic cell in detail. Prokaryotes. Prokaryotes are the organism with a prokaryotic cell which does not have a well-defined nucleus. So in this diagram we can see instead of a well-defined nucleus there is a nucleoid in the center of the cytoplasm. So now we'll study in detail about prokaryotes. Prokaryotes, the nuclear region is poorly developed. They don't have a well-defined nuclear region and the nuclear membrane is absent. Studied in nucleus, the outer membrane, outer double-layered membrane is called as nuclear membrane, but in case of prokaryotes, the nuclear membrane is absent. An undefined nuclear region containing only nucleic acid is called a nucleoid. So in prokaryotes, the undefined nuclear region which is containing only one nucleic acid is called as a nucleoid. It will have a single coiled chromosome and membrane bound cell organelles are absent. In a normal cell there are membrane bound cell organelles, but in a prokaryotic cell membrane bound cell organelles are absent. And prokaryotes are generally small in size, that is from 1 to 10 micrometer, that is very small, 10 raised to minus 9 it is 1 micrometer. So it is only 1 to 10 micrometer size in size. The size of the prokaryote is very small when compared with the eukaryote. And examples for prokaryotes include bacteria, mycoplasma, etc. Now it's about eukaryotes. Eukaryotic cell will be having a well-defined nucleus with nucleolus, chromatin material, everything inside. We can see the structure of a eukaryotic cell here. Eukaryotic cell will have a nuclear region which is well developed. They have a well developed nuclear region and nuclear membrane is present and it contains more than one chromosome. In prokaryotes only single chromosome which is in a coiled form, but in case of eukaryotes there are more than one chromosomes present. Membrane bound cell organelles are present. Membrane bound cell organelles are present in case of eukaryotes. And eukaryotes, when compared with prokaryote, eukaryotes are generally large in size, that is 5 to 100 micrometer. And examples for eukaryote include plant cell, animal cell and cell of human, etc. So eukaryotic cell will have a well-defined nuclear region with a well-developed nuclear membrane. Now we can see the differences between prokaryotes and eukaryotes. You can copy down this in the notebook. So prokaryotes, first point, most prokaryotes are unicellular. Most eukaryotes are multicellular. Prokaryotic cell, when we see, the nucleus is poorly defined due to the absence of nuclear membrane. But eukaryotes, the nucleus is well defined and is surrounded by a nuclear membrane. Nucleolus is absent in case of prokaryote, but in case of eukaryotes, the nucleolus is present. Now in coming to the cell organelles, in prokaryote cell organelles such as plastids, mitochondria, Golgi bodies, etc. are absent. But in eukaryotes, cell organelles such as plastids, mitochondria, Golgi bodies, etc. are present. And in prokaryotes, the examples for prokaryotes include bacteria, blue-green algae, etc. And eukaryotes, fungi, plant and animal cells are eukaryotic cell. Now we will be studying about the next basic component of the cell, cytoplasm, in the coming session. We have studied about the three basic components of cell. Now we have to study the third basic component of cell, that is cytoplasm. Cytoplasm is a jelly-like colorless semi-fluid substance that occurs between the plasma membrane and the nuclear membrane. Plasma membrane forms the outer covering of cell and nuclear membrane forms the outer covering of nucleus. So in between the plasma membrane and the nuclear membrane, there is a jelly-like colorless semi-fluid substance that occurs, that is called as cytoplasm. Cytoplasm, we can see the cytoplasm is like a thick jelly-like substance of the cell. Now about the constituents of cytoplasm. The cytoplasm contains several things. It contains variety of cell organelles. Cell organelles are the structures helps to perform various functions, and other soluble waste products. Some of the waste products also, soluble waste products also will be present in the cytoplasm, and storage products like starch, glycogen, lipids are present in the cytoplasm. So cytoplasm contains variety of cell organelles, soluble waste products, and products like starch, glycogen, lipids, etc. Functions of cytoplasm include, actually the cytoplasm acts as a store of vital chemicals like amino acids, proteins, sugars, vitamins, etc. These are the chemicals which are needed for body building and other major metabolic activities. So the cytoplasm forms the storehouse of vital chemicals like amino acids, proteins, sugars, vitamins, etc. And it also acts as a site of certain metabolic reactions. Various metabolic reactions are the reactions which take place inside our body, and it forms the cytoplasm actually acts as site of these metabolic reactions like synthesis of fatty acid production take place in the cytoplasm, production of nucleotides take place in the cytoplasm, and also it acts as a site for cellular respiration also. So the functions of cytoplasm include act as a store of vital chemicals like amino acids, proteins, sugars, vitamins, etc. and the site of certain metabolic reactions like synthesis of fatty acids, nucleotides, etc. Now about protoplasm. You must have heard of the term protoplasm. Protoplasm is actually cytoplasm together with nucleoplasm. We call it as protoplasm. Cytoplasm is the jelly-like substance which is present in between the plasma membrane and the nuclear membrane, and nucleoplasm is the jelly-like substance which is present inside the nucleus. So protoplasm means cytoplasm together with nucleoplasm. We call it as protoplasm. Now about the cell organelles which are present inside the cytoplasm. There are various organelles with various structures are present in the cytoplasm. Organelles are the small solid structures present in the cytoplasm which perform different functions. And the major cell organelles, these cell organelles will have different functions to perform. The major cell organelles are endoplasmic reticulum, Golgi apparatus, lysosomes, mitochondria, plastids and vacuoles. So we will be studying about these 6 cell organelles in detail in this chapter. So they are endoplasmic reticulum, Golgi apparatus, lysosomes, mitochondria, plastids and vacuoles. So first we will be starting with the first cell organelle, that is endoplasmic reticulum. We studied about the cell organelles and 6 different types of cell organelles which we have to study in this chapter also we studied. That include endoplasmic reticulum, Golgi apparatus, lysosomes, mitochondria, plastids and vacuole. So we will start with the first cell organelle, that is endoplasmic reticulum. Endoplasmic reticulum, I think you might be hearing this term for the first time, right? Endoplasmic reticulum. Endoplasmic reticulum, these structures are called as endoplasmic reticulum. Okay, when you draw the diagram of cell, I will not draw the entire diagram of cell here after. I will be drawing the structure of each and every organelle separately, but when you draw the cell, you have to include all that organelles inside your diagram. So this will be the structure of endoplasmic reticulum. So we will see the endoplasmic reticulum, structure of endoplasmic reticulum. Now, it's actually a large network of membrane bound tubes and sheets. It forms a large network of membrane bound. It's actually made up of membrane bound tubes and sheets. And it look like long tubules. It may look like long tubules, or it may look like round or oblong bags or vesicles. So it may look like long tubules, or it may look like round or oblong bags. It will have a similar structure like plasma membrane itself. Plasma membrane is made up of proteins and lipids. We studied that when we studied about the plasma membrane yesterday. So this endoplasmic reticulum is also having the same structure like plasma membrane. So this is about the structure. It's actually made up of membrane bound tubes and sheets, look like long tubules or round or oblong bags. Now we have to see the types of endoplasmic reticulum. There are two different types of endoplasmic reticulum. That is rough endoplasmic. ER is the short form for endoplasmic reticulum. Don't get confused, okay? ER is endoplasmic reticulum. So rough endoplasmic reticulum and smooth endoplasmic reticulum. Can you tell from the name itself, rough endoplasmic reticulum and smooth endoplasmic reticulum, what may be the difference of them? Rough endoplasmic reticulum may be rough and smooth endoplasmic reticulum may be smooth. Roughness and smoothness in the appearance. Here, see, we can see the rough endoplasmic reticulum looks rough under microscope. When we view it under the microscope, this rough endoplasmic reticulum will have a rough appearance. But smooth endoplasmic reticulum will have a smooth appearance when we view it under the microscope. What may be the reason for that? Actually, there are certain particles called as ribosomes. Ribosomes, we will be studying about the ribosomes. So there are certain particles called as ribosomes are attached to the rough endoplasmic reticulum. If these ribosomes are attached, it will have a rough appearance. They are actually, see, this small round spherical bodies called as ribosomes. So if the ribosomes are attached to it, it will have a rough appearance and hence called as rough endoplasmic reticulum. In smooth endoplasmic reticulum, these ribosomes are absent. That is the main difference between rough and smooth endoplasmic reticulum. So now we will study about this rough and smooth endoplasmic reticulum in detail. We will see the rough endoplasmic reticulum and smooth endoplasmic reticulum in detail now. So this is rough endoplasmic reticulum. It will be mostly situated near to the nucleus, so that helps in the synthesis of various components and also helps in the transport of various components. So rough endoplasmic reticulum will be having ribosomes attached to it, and because of the presence of the ribosomes, they have a rough appearance. And smooth endoplasmic reticulum will be having oblong or long tubule shape, and they will not have the ribosomes attached to it. Ribosomes are absent. Now these ribosomes, we studied ribosomes are some particles which are attached to the rough endoplasmic reticulum, and these ribosomes are site of protein synthesis. Synthesis means production. So ribosomes are the site of protein synthesis. Very important. The site of protein synthesis is ribosomes. So the ribosomes are the site of protein synthesis, and protein synthesized is sent to various places of the cell using endoplasmic reticulum. They are actually like a network. So the protein which is synthesized by the ribosome will be sent to various parts of the cell using endoplasmic reticulum. Endoplasmic reticulum helps in the transport of the protein which is synthesized. And this smooth endoplasmic reticulum helps in the manufacture of fat molecules or the lipids. They help in the manufacture of lipids. So you might have heard of protein and lipids. We studied plasma membrane is made up of two constituents, proteins and lipids. So these proteins and lipids produced by the endoplasmic reticulum helps in the formation of the membrane. And the process of formation of membrane is called as membrane biogenesis. Remember, rough endoplasmic reticulum or the RER helps in the synthesis of protein, and smooth endoplasmic reticulum helps in the manufacture of lipids. These proteins and lipids can together make membrane, and the formation of membrane by proteins and lipids is called as membrane biogenesis. Another important topic, membrane biogenesis, that is the formation of membrane, nothing. Membrane biogenesis means the formation of membrane. Now, the sum of these proteins and lipids can be converted to form hormones and enzymes. Some of the proteins and lipids produced by the ribosomes can be used for the formation of hormones and enzymes, etc. So, this is all about the endoplasmic reticulum. Now, we have to study the functions, main functions of endoplasmic reticulum now. Let's see the functions of endoplasmic reticulum. The main functions of endoplasmic reticulum. Actually, this endoplasmic reticulum acts as a network system. It always forms a network system inside the cell cytoplasm, so that it helps to serve as a channel of transport of materials within the cytoplasm. Within the cytoplasm, it helps in the transport of materials. It acts as a channel for transport. So that is the first function of endoplasmic reticulum. It always forms a network system, hence it acts as a channel for transport of materials. Second function is it forms a cytoplasmic framework providing site for various biochemical activities. One of the biochemical activity we know, that is protein synthesis, that take place in the endoplasmic reticulum. So these act as a cytoplasmic framework providing site for various biochemical activities. And the third function is in liver cells, the smooth endoplasmic reticulum, SCR is smooth endoplasmic reticulum. In liver cells, smooth endoplasmic reticulum helps in detoxifying various poisons and drugs. The poisons and drugs which is entering our body can be detoxified by this smooth endoplasmic reticulum, which is present in the liver cells. So these are the three main functions of endoplasmic reticulum. One is helps as a channel for the transport of materials within the cytoplasm, within the various regions of the cell. And second, it forms a cytoplasmic framework providing site of biochemical activities. And third is in liver cells, smooth endoplasmic reticulum helps in the detoxification of poisons and drugs. So that's all for today. Today we will conclude with today's topic. Tomorrow we will study the remaining cell organelles, Golgi apparatus, lysosomes, mitochondria, plastids and vacuoles. Thank you.